I'm Dr. Rana Eldanaf, and I'm a research scientist in Dr. Claude Despin's lab at NYU Abu Dhabi. In neuroscience, it's very important to understand the vast diversity of neurons that make up our brains. And the research in the Despin lab is essential to gain insight into how these neural structures are formed and how these could be compromised in disease and in neurodegenerative disorders. We use the Drosophila fruit fly, which provides a powerful model organism to understand various aspects of neural circuits development, function, organization, and disease. The fruit fly has a relatively simple brain structure, yet it allows for very sophisticated behaviors. The main questions that we try to answer in our lab are, first, what are the mechanisms involved in stem cell division to generate the vast diversity of neurons that we see in the brain? Second, how do neural circuits form during development in order to establish complex neuronal connections? To answer these questions, we use the latest technologies in live imaging, transcriptome analysis, and genetic manipulations in order to selectively label and manipulate distinct neuronal populations. Both of the Despin lab groups at NYU New York and NYU Abu Dhabi work together to create a developmental atlas of gene expression in neurons using machine learning and gene sequencing to categorize more than 250,000 neurons in the fruit fly brains across development. These results were recently published in the scientific journal Nature. And more recently, the Despin lab started studying the red palm weevil, a deadly beetle that attacks more than 50 species of red palm trees. And it has already wiped out hundreds of millions of dollars of red palm trees across the Mediterranean. In collaboration with researchers at Khalifa Center for Genetic Engineering and Biotechnology, our research aims to gain insight into the red palm weevil brain organization and gene regulations in order to direct pest regulation efforts by introducing genetic manipulations in the species. Under the Neural Systems Research Program, a collaborative effort between Dipesh Chaudhry and Justin Blau's lab is working to generate single-cell expression profiles of mouse pacemaker neurons at different times of the day to understand the mechanisms driving rhythmic cycles. 24-hour pacemaker neurons provide a powerful tool to understand how the transcriptional state of a neuron is translated into rhythmic activity and ultimately into behavior. And the Fischl group at NYUAD helped in the establishment of a viral core that produces both recombinant adeno-associated viruses and recombinant rabies viruses. Studying the role of specific neuronal subtypes in disease and development has been limited to the use of transgenic animals, so these tools help extend that into non-genetically amenable species. The pipeline has been standardized to ensure the production of reliable, low-cost, high-quality reagents, which constitute an essential support to the research of the scientific community.